It's my pleasure to introduce Dr. Violante from the University of Surrey, and she also uses fMRI a lot with neuromodulation, but has a focus on transcranial electric stimulation. I will present some of her research in that area to us.
I want to thank Alex and Ivan for the workshop. It's been really great, and I've learned a lot, and it's really nice to see that so many people are using similar techniques and exploring similar stuff. Okay, so we are particularly interested in using brain stimulation as a way to modulate cognitive function. So we know there's some relationship between these complex networks in the brain and behavioral function. And our hopes, as many others, is to introduce some sort of external modulator that will have an impact in brain dynamics and modulate behavior. And as others, we've been really interested in brain stimulation, particularly electrical stimulation, as a way of causally investigate brain behavior relationships, but also as a possible avenue of therapy for a variety of neurological and psychiatric disorders. And talks both yesterday and today touched on that, and why this will be such a promising technique. So it's easy to use, has a good safety profile, and particularly when talking about electrical brain stimulation, current brain stimulation like TDCS and TACS type of tools, we're talking about relatively cheap techniques. However, as many of you touched both yesterday and today, there's controversy around replicability. And we know that behavior effects are highly variable. And the reasons for that are poorly understood. So we have some good ideas of why that's the case, but we don't know exactly what causes, what will generate an effect versus what we want. So in a way, a lot of the issues in the field with poor replicability have to do with sort of a traditional approach that people used a lot for TDCS and TACS, which is to define a region a priori and define frequencies and phase intensities and use this in a population to bring people in the lab, look at behavior, do some control conditions, and gather some results out of this. And as many of you touched on already today, there's challenges with this. So one has to do with verifiability. So we don't know whether we indeed target the networks or the regions that we set up to target in the beginning. And this is hard to do without combining with imaging, which I don't need to explain much to this crowd. The second has to do, and this is the biggest one, is to do with interindividual variability, right? And this is the sort of like one approach, one fits all approach has been shown to have a lot of issues. So we've been seeing that stimulation parameters, they may vary across subjects, and this has to do with interindividual variations in the neurobiology of the brain. And when we talk about subjects with brain pathology, this is an even bigger challenge. And third challenge here, which has to do with specificity and a lot of very interesting computational modeling here addressing focality. And another issue has to do with our limited capacity to target deep brain regions. And today, great talks by Joel Marks saying like how you can target a cortical region and through that target the hippocampus, but doing this in an indirect way. So what we've been doing a lot is combining, so to address this first challenge of verifiability is to combine brain simulation with MRI. And you've heard about all the advantages for this. So one is the absence of artifacts that we have quite abundantly on EEG recordings, because it's just an order of magnitude larger what you can record from the brain on this microvolt range and what you input with electrical stimulation in the millivolt range. The other advantage has to do with a high spatial resolution, so you can look at cortical and subcortical areas and you can really map this large-scale networks. And also touched on yesterday, another advantage is that you can combine additional modalities. So once you have a participant in the scanner, it's not that much more expensive to acquire spectroscopy, for example, to give you an idea of some metabolites in particular regions, acquiring DTI to give you an idea of white matter tracks. And we've also heard about some cool techniques that might tell you a bit more about where the current is spreading. Of course, there are issues here. So this is a voltage surrogate signal of neuroactivity. And as we just heard, it has a low temporal resolution, so you can only really look at slow oscillations. Okay. So I just wanted to talk a little bit about one of the studies that we did some time ago. We wanted to target cognitive function, and particularly we wanted to target working memory. And it is very well established that working memory is based on this frontal parietal network. And it's also well established the importance of theta oscillations for working memory processes. So we got people to the lab and asked them to do different tasks, different working memory tasks that vary in difficulty, if you want. So in working memory load. So we had a very easy task, very controlled task, whereby participants are shown an arrow that points to the left or to the right. And they just have to press a button that matches the direction of the arrow. And then we did again these classical tasks that looked at working memory. We did a 1-back and a 2-back task. And we combined this with tags to the frontal and parietal regions. We use 6 Hertz frequency and 1 milliamp intensity. And we did this in 2 conditions. One in which the current that we input to here, F4 and P4, was synchronous, so 0-degree phase lag between it, and the other, it was 180 degrees between the current that we input to F4 and to P4. So we look at how participants responded to stimulation in different tasks. So what I'm showing here are reaction times for these tasks. And what we observe is that we only see modulations in behavior when we look at this 2-back task, which is the hardest task. And what we see is that during this synchronous condition, participants got faster in this task compared to the 118 to the sham condition. And interestingly here, they got as fast as they were for the 1-back, which is a much easier task. But we are back to this issue, right? I wanted to target the frontal parietal network, but I have no idea if that's indeed what I did. So to address this, we got another group of participants in the scanner, and we did stimulation while participants would do this task. And we did this in a block design, so very simple block design type of task. You have your 2-back fixation for rest, and this allowed us to have blocks of the task with stimulation and blocks of the task without stimulation. So this allows us to compare, to contrast what's happening during and without stimulation while participants are doing the same task. And we had like 2 main hypotheses here. One would be that tasks would modulate activity and current activity across these large scale networks. And the second will be that this modulation will be dependent on the type of stimulation that we're inputting, the parameters of stimulation that we're inputting. So looking at what happened with this 2-back task, what we observed is during this synchronous condition, participants got increased brain activity in this frontal parietal network. So this is while they're doing the 2-back task, synchronous versus sham condition. Interestingly, when we look at what happened during the synchronous condition, we see a very different effect to sham. So we see modulations on default mode network. So we show that indeed there's modulation of brain activity, and this is dependent on the stimulation condition that we use. Then the second hypothesis had to do whether we also would see effects in brain connectivity. And really nice effects in brain connectivity have been shown with TMS here today. So we also look at this in terms of tasks. And indeed, that was the case. So when we applied the synchronous simulation, we looked at the connectivity between the parietal cortex, so underneath the electrode in the parietal cortex, and we see an increase in connectivity with frontal areas. And during the synchronous condition, we see something very different. So when you look at the same seed region, so this parietal region, we see modulations of connectivity, so increases in connectivity, but with the occipital cortex. So very different patterns depending on the stimulation parameters. Interestingly, we did this also with this control tasks, this choice reaction task. So same participants, same session in the scanner. And here, this task, we already showed no effects in behavior, but also here we show that indeed this network that we're trying to target is not very involved in this particular task, and we don't seem to see a difference in activity between the different simulation conditions for this control task. And also, we didn't see any modulation of connectivity compared to sham for this control task. So just a bit reiterating that how important brain state is for when we're trying to manipulate brain networks, the activity of brain networks. Cool. So I was talking, the second challenge that I said in the beginning, and many of you talked about, has to do with interindividual variability. And this is the biggest challenge to have to apply brain simulation in the clinic, right? And we know that there's like many sources of interindividual variability, structural, neurochemistry, genetics, age, gender, so on and so forth. And the lab, when I started this brain simulation studies, they're quite interested in traumatic brain injury. And in TBI, two of the things that we know is that as a consequence of the injury, there is structural network disconnection, and you can see this in white matter tracks. And there's also consequent alterations in functional connectivity. And there are also metabolic changes. So these were two things that we started by looking at. So these sources of variability in patients and whether they were related to the effects of brain simulation. So I'm just going to focus on the white matter tracks today. And, sorry. Okay. And to focus on that, I want to talk about a different study. In here we use TDCS, and we use a different task. So we're using a stop signal task. This is a task typically used to study inhibition. And in this task, participants see a fixation cross, and then they see an arrow that points either to the right or to the left. And this happens in the majority of trials. And then in a smaller percentage of trials, they get a stop signal. And the stop signal tells them that they should inhibit their movement and do not press a button. And the lab that I was at showed that patients with traumatic brain injury have difficulties in this task. They had shown before that in particular, this has been associated with a salience network that involve regions of the inferior frontal gyres and the anterior insula. So we got participants in the lab to do this task in the scanner while they received TDCS in 3 conditions, so either cathodal, anodal, and sham. And interestingly, we also did this in healthy controls, and in the healthy controls, we observed an improvement in the task, in the stop signal task, but we don't see this in patients. So we then were able to look at what could be sources of variability? So what could be leading to this difference between patients and controls? So in the paper, there's a bit more on like bold activity and how the activity in different networks are modulated depending on different stimulation conditions. But here I just want to focus on white matter tracks. So we looked at, so we had these patients in the scanner, so we could collect DTI data as well. So we looked at, in particular, 2 tracks that connected the area that we aiming to stimulate to this pre-SMA track. And the reason we looked at this is because previous work in the lab has shown that this particular track was related to how well people perform this stop signal task, particularly in patients. And we know that, and we just show that, well, patients have decreased fractional anisotropy compared to healthy controls in this tract, which we expect. And this is also true for the cingulum tract and as well for the whole skeleton. Importantly, when we look at how patients and controls perform this task in particular conditions, and here I'm showing the nodal condition, we see a correlation between the fractional anisotropy in this tract and the SSRT, which is a measure of inhibition in this task. And we see this for patients and controls for this particular track. And in the patients, we also see a correlation between fractional anisotropy and the SSRT. So in essence, well, what I want to show with this slide is that the response to non-invasive brain stimulation following brain injury is dependent on the structure of the brain network stimulated. And in that case, in this paper, we show a nice dissociation between those that have intact or more intact tracks compared to those that don't, and whether they would respond to brain stimulation or not. So those that had better fractional or higher fractional anisotropy benefited from another stimulation, whether those that did not benefit from stimulation. Okay, so continuing on this idea of interindividual variability, and I touched on many of those, but then we also have other issues, right? So what frequency does one stimulate? If you have more than 2 electrodes, at what phase do you do this? And we start building a big matrix of parameters, but then we can also ask what intensity we stimulate. And what you can see from here is that, well, you have this combinatorial explosion of possibilities. And clearly, one cannot explore those in a systematic way. So what we're trying to do now in the lab is employing neuroadaptive paradigms, so particularly Bayesian optimization, to try to help us understanding which parameters better modulate networks at the individual level. So the advantage here of Bayesian optimization as other active learning paradigms is that the algorithm learns in real time, and you can adjust this to the individual. And Bayesian optimization is particularly useful when data acquisition comes at a cost. And here you have many costs. So one is time, your participants' time, comfort. If you want to do this in the scanner and target different networks, scanning costs as well, and also the amount of stimulation that you can give per session if you wanted to try this in a sort of more systematic approach. Indeed, Bayesian optimization approaches are used for in our field, and they have been recently applied to invasive stimulation protocols. Okay, so what's the idea here? So in a nutshell, we can think of brain stimulation as this 2-stage iterative loop that repeats itself. So the first stage is a data modeling. And what we do here is that we use a probabilistic surrogate model. So the idea of the surrogate model is to estimate the objective function. So the objective function is what we would like to know, is this relationship between particular stimulation parameters and brain response. And that's unknown, but we can estimate that. And typically this is done using Gaussian process regression, which will give you across the whole space an idea of how well will give you an idea of this object, will model this objective function across the whole space, so across the whole big parameter space. And the second stage is a guided search or a proposed point, and for this we use an acquisition function. So the objective of the acquisition function is to take the information from the objective function, so from the posteriors of the objective function, and use it to propose a point in the parameter space for which to sample next. And the acquisition function has to balance this trade-off between exploration and exploitation. So you want to explore your parameter space enough, and you want to start to exploit it when you think that you are close to your maximum. Okay. All right. So to show how this could be applied to brain stimulation, we started with a simple proof of concept study. And to do this, we use something that is very easily evoked by tags, which are phosphines. So these are visual percepts. They are induced because the current passes through the retina, and they're really nice because people have been reporting this for a long time. So there's studies since the 60s and the 70s showing a particular function, response function between the frequency of the stimulation that you impose and how noticeable those phosphines are. Okay, so we use this adaptive Bayesian optimization paradigm, and what we ask our participants to do across different montages, I'll show in a bit, was to just basically tell us if they perceive stimulation, if they perceive the phosphines stronger, by this we meant like brighter, or occupying more space in their visual field in one block compared to the other. And what so we do this in a closed loop, and what's happening is that we're using this binary preference to update our model, and what we see is that the variance across the mean is decreasing as the iterations increase, and we are approaching our optimum. Okay, so in this particular case, we had 3 different montages. So we started by the montage that has been used previously in the literature. So one electrode in CZ, another in OZ. So this 1D is an easier optimization problem. And what you see here in the black dots are dots is where the samples, that the algorithm proposed and were measured, and the darker blue colors showed where the phosphines were perceived as brighter, so higher phosphine perception. And we show this nicely replicated previous results, and we also repeated this with different montages where we have like different stimulators that were inputting stimulation at different frequencies and phases. And interestingly, here again, you can see all the points that were sampled and where we obtained the maximum for phosphines perception in the darker blue color. And what we see is that the preferred frequency was very similar to what we obtained here, so around 19 Hertz. But the preferred phase was an in-phase condition. So from this slide, just to show that we can combine Bayesian optimization with electrical stimulation, and this works on an individual basis. For each of this montage, it took us about 5 minutes to go through this big parameter space, compared to what it take about 80 or 90 minutes to do this in a more systematic way if we sample the same points. And to the phase effects, computational modeling here, I really like this picture shows us that, well, the current is passing through the retina, and the phosphine's intensity is higher at this in-phase condition for both this O1, O2, and F4, P4 montage, because there's some sort of stimulation of the current. So, and whereas in the 180 we see interference. Cool. So what we want to do now, and what we are pursuing now in the lab, is to do this in what we actually want to modulate, which is brain networks. So we're starting by focusing on frontoparietal networks and this dynamics between frontoparietal and default mode networks. So the idea here is that we get participants in the scanner, put electrodes in particular positions. We're acquiring real-time FMRI. We extract these brain networks per subject, and we model. So we have an information from our objective function that is used in our acquisition function to propose a next point to sample. And we hope by doing this in this closed loop iterative approach will allow us to define optimal parameters to stimulate for each individual. Okay, so just some conclusions of what I said today. So the first one, the importance of combining NIBS with imaging, and how important this is to address verifiability. And as many of you showed today, MRI is a powerful approach to do this. It offers high spatial resolution. You can use complementary modalities, and you can get this without artifacts, major artifacts. And this is particularly important in patient populations where you might expect a larger variability. And then related to this interindividual variability, the way we're trying to address this is use this neuroadaptive stimulation approaches, so closed loop neuroadaptive approaches. And we think that this would be hopefully a fast approach and a reliable approach to identify ideal parameters in local that we can define for different networks, depending on what we want our targets to be, so where we want to modulate activity or connectivity. And we think this is particularly helpful to explore many more experimental conditions than we currently can with a standard methodology. And then for specificity, many of you talked about how computational models can help us to define targets, and we show really elegant approaches to improve specificity using modeling. And also thinking about how targeting deep brain regions, perhaps thinking about novel approaches that might be able to target deep brain regions like the hippocampus in a direct instead of indirect way. And just to acknowledge a lot of people that contribute to the work across a number of universities and the funding bodies as well. Thank you.
Thank you, Ines, for the exciting talk. We have a number of questions. First, did you compare online and offline FMRI results? Probably that's related to possible TES-induced artifacts.
Oh, I see. So for, well, yes and no. For this particular experiment, we designed it in a very different way for the offline and the online. So for the offline, people would come in 3 different days. They would have stimulation for the whole duration of the task, which is about 12 minutes per task. For the FMRI, we weren't powered to do this. So we opted for a different design where we had less than a third of the trials that we had for the behavioral experiments. So we were really looking to see whether we could modulate network activity. So we changed this. Our stimulation during the FMRI is just 30 seconds. And we have here, if I'm not mistaken, 6 blocks with stimulation and 6 blocks without stimulation. We do see some correlations between reaction time and increased bold activity in the frontoparietal area. So when we looked at we looked at particular bold signal in the regions underneath the electrodes that we stimulated. And in that case, yeah, for the parietal one, we see that higher bold activity in the right parietal area underneath the electrode was correlated with faster reaction times. So mix.
Thank you. What are your thoughts on analyzing FMRI during TES compared to pre-post design?
So we never actually did a pre-post design, so I can't talk by experience how different it would be here, for example. The reason that that I always opted for during is that, well, one, we can, and we're expecting these effects to be sort of transient, particularly with TACS. Not that there is a great body of literature exploring how those effects. So I always thought that would be a best approach to just stimulate doing a particular task. And I think one thing that we show with TACS and also with TACS and then with TDCS is that it's really important what the participant is doing. So if you target the same areas of the brain, exactly the same parameters, but they are doing a different task, we don't see behavioral effects and we don't see modulations on the network. So I would say that from our limited experience, targeting during the task that you want to modulate is what makes more sense, where we see the greatest effects.
Okay, very related questions, but maybe you can answer it as well. How do you handle carryover effects from one condition to the next in closed loop optimization?
Brilliant question. I don't know. So again, those effects, right? So what we're assuming is that if we stimulate for 32 seconds and then we have a resting period between stimulations of the same order of time, so about 30 seconds, we'll minimize carryover effects. If this is entirely true or not, I don't know. So we looked at our FMRI results and we tried to look at carryover effects, and we don't seem to see this, but this might be because we're not powered to see those. I also tried to look this on EEG and with TACS, so not recording during or recording during but not being able to recover the signal. When we stimulated for about 30 seconds, we seem to see an effect, in this case on theta activity, 1 to 2 seconds after, and then it just goes away. So this just talking about power in theta band. So yeah, I don't know. I think more studies would be needed for this. I'm not super worried about it, because what happens, the advantage of Bayesian optimization here is that you are exploring this space, and we choose acquisition functions that try to balance this trade-off between exploration and exploitation. So if we're getting to a maximum, what is going to happen towards the end of the experiment is that the acquisition function keeps proposing the same points to sample next. So we'll have, like, for each participant, we're expecting this to be slightly different, and we'll have some sort of like inner replication within the experiment.
Okay, and maybe the final question, can you disclose any secret how to record clean FMRI during TES for people who will try to repeat your efforts?
Yeah, sure. So we use commercial, so we'll be using the Neurocon. Yeah, so these are like the Neurocon stimulators. The only thing that we did that wasn't recommended by them was we created a filter that we've put in the penetration panel. I can put it bigger. So so the way they sold this is that you had like an Ethernet cable that goes to the waveguide, and I had lots of issues with this. I realized if I wiggled the cable, if I didn't put it like exactly like they wanted and very flat, I would have noise in my images. So we opted by building a filter that goes Ethernet to the penetration panel and then penetration panel to Ethernet. That worked really well for us. And I know that their new approach now, Neurocon, is to build this, is to offer these filters. The other thing that we do is we always use, we always look very carefully to our images in this pre-processing stage. So we're using FIX, which is an ICA-based algorithm that labels different components in signal and noise. I don't see particular artifacts that I would associate with electrical stimulation that I don't see in other recordings. There might be something that I'm missing here, but everything that I've done in terms of phantoms and we don't see particular artifacts, and definitely we don't see artifacts that would explain these differences, because we always use then control conditions, and if the artifacts were consistent, they will be there in the control condition.